Thank you, Alice. I would like to welcome you also all to the last lecture of the series for the 2017. We will resume the nano lecture series in the fall. I think the first lecture will be in October, and this lecture series is part of the NIHS Nanotechnology Health Implication Research Consortium, which was established last year. We are honored and pleased to have with us today Wendel Wohlleben from BASF. We know him for quite some time. He's a collaborator here at our center and friend. I won't go through his extensive CV. He's the senior scientist at BASF and Department of Material Physics and Department of Experimental Toxicology and Ecology. He's leading a lot of projects in Europe in nanomaterial and nanosafety research in general, so he's well known in the field. So we are really pleased to have him with us today. And I think the theme of the presentation also is very interesting and important for nanosafety research. So it's a grouping of nanomaterials. It's I think the Europeans are ahead of us when it comes to this advanced strategies for screening nanomaterials for nanosafety research. So it's and I'm very pleased to see that this the grouping, which is based on the chemical properties of physicochemical properties of materials, is taking it to the next level. So in addition of the extrinsic properties, now we're looking at how to include in the grouping strategies lifecycle transformations and other things, which I think that will make this grouping approach a very useful tool for our attempts to narrow down the gazillion combinations of screening for nanosafety research, which is always a challenge. And I think without further ado, welcome Wendel, and we look forward to your presentation. Thank you.
Alice, should we have to handle? And so I will start by a general introduction into our grouping schemes and how they compare to others, also in the regulatory perspective. And I will then show you very specific examples of methods that we develop in our labs in collaborative projects about extrinsic properties. I'll show you what that means and how we use them to assess similarity of materials. And then finally, we'll see how we can expand this, like Phil said, to a next level of complexity to nano-enabled products, about the life cycle of these. So earlier this year, Debbie Kaiser and others from NIST published this book about metrology and standardization of nanotechnology. And I wrote a chapter together with collaborators about applications of nanomaterials. And so this is a very, very simple example. It just shows you how a nanoscale silica can introduce a stabilization by changing the van der Waals interactions between these sodium chloride crystals. So this is the famous flow effect, or the rheology modifier effect of silica additive in food. It just keeps salts apart from each other. In that case, it's salt crystals, could be other particulates. And so you see here the naked sodium chloride crystal. Here you see how the surface is coated by the specifically fluffy aggregated silica aggregates. And this is the effect. Without the silica, this would be the way that your salt looks like. and with the silica, it's a nice flowable powder. So it's a very simple application, which is not new at all, has been around for some time, but I'm glad that we can communicate in this way about these applications. Here's another example, again, based on particles. If you use LCD screens, also this beamer uses this technology, you have pixels which have a certain color, and the liquid crystal technology is just there to shut it on or off, so to modulate the intensity. But in order to modulate the color, you need color filters. And today's standard, the best we have is a 6,000 ray on-off ratio. So that's a very, very clean, bright color. This is because it was launched in 2013. We have an diketopyrrolopyrrol, so that's an organic compound, which we engineer to have a very specific particle size, a very specific particle surface, and a very specific shape. And only then we achieve the specific color and contrast effect, so to suppress any scattered light. Again, earlier versions of this technology have been around. So the previous generation was in 2005, and before that in 1997. And you see how we gradually increase the brilliance, the color depth of these materials. So these are particle-based systems. And now the question is, how do these particles potentially have an impact on the safety of their use? And many steps contribute to the final adverse outcome. First, of course, these materials have certain properties, which may be even different after they are released from a powder, a suspension, or a matrix. So we need to know something about their exposure. We need to know how they are even available. Then they have to cross certain barriers in order to get to any place where they could have an adverse action. And only then we will have biophysical interactions, maybe cytotoxicity or other inflammatory triggers, and then finally an adverse effect. Our thinking was that each of these steps could provide a property, a lever to compare between different materials. So a similarity in the way that they are dispersed, a similarity in the way that they cross barriers, or a similarity in cellular effects. All of these can be used to compare from one material to another. And this is what we do in grouping and read across. And so this book from 2 years ago was the first that took really a life cycle perspective on nanomaterials, and it shows some first examples of grouping. But now grouping has advanced. It even is part of regulatory decision-making concepts. And we have two concepts that we need to look at. First, in the top, of course, we need to ensure that new nanomaterials that are being developed are safe at the time that they are brought to the market. So this is about novel, potentially unknown materials, and we want to compare to known materials so that we can narrow down the uncertainty about the potential effects of these materials. But there's another application for grouping and read across, which in fact for a company like BASF, which produces many nanomaterials, is even more important. And that's the re-evaluation of conventional nanomaterials, such as those silicates and pigments that are presented on the first two slides. These materials are not necessarily novel. They may not be as exciting and sexy as quantum dots and carbon nanotubes, but they are produced in large quantities, and regulators are interested in them again now, because now we know that due to their particle structure, they may have a specific action. So these are the big guiding questions about grouping and read across. How do we compare between different nanomaterials or between different forms of one and the same substance? How do we compare between a nano form of a substance and a bulk form? And I'll explain the word nanoform in a second. And how do we compare between different nano-enabled products? Now, the word nanoform and discrete form, that is a regulatory term. The European Chemicals Agency, ECHA, has published a best practice document which introduces the terminology nanoform. And nanoforms, so if we stick to the example of the pigments, could have different sizes. So one that has 20 nanometers would be a different nanoform from one that has maybe 80 nanometers. But also they could be different in their shape or in their surface chemistry. So these are three core properties that I think everybody in the community would agree on, that these are relevant for a safety assessment, and they differentiate different nanoforms. EPA has something similar. EPA has published a reporting scheme which will probably come into action soon, and they differentiate what they call discrete forms, which is very similar but not identical. So discrete form is defined by morphology or a coating or different descriptors of size. So there's a good overlap with the nanoforms, but it's not identical because they also look at changes in the process. They define 7 standard deviations as a metric to have certainty that one form is really different from another form. And then they also introduce more properties such as charge, surface area, dispersion, stability, surface reactivity. So based on these descriptors, we now have divided the landscape of nanomaterials into categories, which we call nanoform or discrete forms. Now we have to compare these. And I think a leading concept that really has paved the way was published 2012 from 4 NIOSH authors. And they put up a scheme based on a mode of action. So they differentiate, they differentiate high solubility particles against poor soluble low tox, against poor soluble high tox, against fibrous particles. And they introduced some very important concepts which we took and further developed for our grouping schemes. They introduced the importance of benchmark materials to differentiate the mode of action which will guide the assessment for these different groups. You will assess a soluble material differently from a fibrous material. They highlight that the dose metric may be different for these different groups. And this is now mirrored in the grouping concept or the grouping framework that we developed under the frame of ECETOC, the European Center for Toxicology and Ecology. It was published 2015 and follow-up 16. We have a tier 0. These are the different nano definitions. I'm not going to talk about this today because it is a topic of itself, but let's just assume that we have identified a material as being a nanomaterial. Tier 1 is oriented along these 4 groups from the NIOSH authors. So we sort out materials that are highly soluble. We sort out high aspect ratio materials from the morphology, and we sort out those that have a known toxic component via the composition. And in the paper, we give quantitative cutoffs for each of these parameters. So this defines which information needs we might have, and we fill these needs by system-dependent or extrinsic properties, in the sense of properties that depend on the interaction between a particle and its environment. We believe that these properties are key to understand the different groups, and so we will have 3 main groups: the solubles, the high aspect ratios, the passive nanomaterials, which are negative by all these properties, and then probably different subgroups of active nanomaterials. And all of this will support risk assessment. If we don't come to an end, if these properties are not enough, we can still go to in vivo testing. That's our tier 3. So that's our last resort. And if also in vivo testing does not allow us to identify similar materials, then we will have a case-by-case assessment, which will be guided, which will be very targeted by all the information we have gathered along the way. Today, I will walk you through different elements of this framework, and I'll show you specifically how we think it can be done. And to that end, I'll first list again the properties that we use here. intrinsic properties oriented along the 4 groups originally proposed from NIOSH. Then our system-dependent properties, our use, release, exposure assessment, biopersistent, and in vitro effects. We use all of these in tier 2. There are some things we do not use for a good reason, because they don't help us to refine the grouping with those test materials that we look at. That might change in future, but up to now, we don't need size, hydrophobicity, surface charge to look at the groupings in detail. We still come to correct groupings without these properties. And that also applies to crystallinity and to corona formation. Just a short word on crystallinity. We all agree that you want to know which crystallinity your material has, and we do this as part of our composition identification. So of course we want to be able to say this is a rutile or this is an anatase material, but for grouping, many people use this as a proxy to estimate the surface reactivity. Well, instead we just measure the surface reactivity, because this is much more direct measurement of the actual mode of action than assessing it indirectly from crystallinity. And that's why we don't use crystallinity to define similarity between materials. Let me show you one example how this looks like before we go into details. So here we have a couple of metal oxides that are well known, many of them from the OECD sponsorship program. And so we first look at our tier 1 assessment. Let's start with zinc oxide. Of course, we know zinc can be cytotoxic. And actually, in tier 1, zinc oxide is not water soluble above our cutoff. So we do not consider it as water soluble in tier 1. But then if we go to a dedicated dissolution setup, and we'll talk about this in a second, in tier 2, we do find that it dissolves in pulmonary conditions quickly. This is what you would really expect from in vivo. And so this material goes to an assessment mostly predominantly via the solubility, the soluble effects, and that's confirmed in vivo studies, short-term inhalation. Copper oxide, same story, goes to the soluble material assessment. Titania, different. It only has cellular effects, but there are effects, so we group it as one of the active materials. For the iron oxide, it goes to the passive materials because none of the properties is above the cutoff. For the cerium dioxide, again, we have cellular effects, and actually with our refined surface activity, we have effects there too. So the cerium dioxides, both of them go to the group of active materials, and again, that's confirmed by our in vivo data. Finally, barium sulfate. Barium is on our list that is above the trigger for the composition, but then it turns out nothing else happens, no reactivity, no cytotoxic effects, so it still ends up in the group of passive materials. This is how we would do it in our tier 2 assessment, and of course, one would still have to look in detail how to compare between these active materials. To give you a summary of the in vivo comparison that we made here, so for all of these materials, short-term inhalation studies have been performed, and we have 0 false negatives. That's very important, of course, for us, that it is a conservative grouping, so we do not underestimate the hazard. But we do have 3 false positives from different identifiers. So for instance, we see some mobility for the phosphate, we see some activity here for graphite or for one of the pigments, which in vivo did not confirm. So this is, in terms of accuracy, quite okay, quite acceptable for us. And yet we know that not everybody would agree with the properties that we have chosen. So this is a slide I stole from Gemma Jana from the Guide Nano project. She presented it at the nano-EH in Denmark. And she highlights that apart from our ECETOC proposal, of course, there was also a very, very detailed proposal from RIVM, so the Dutch regulatory labs together with JSC and ECHA, and they have some additional properties on their list which we think are maybe not so essential, but there is also good overlap. Having a what they are chemical identification, what they are physical identification, that is a good overlap, except that we don't use surface area here, but that's okay. And then a description of where they go and what they do. And we think most of these properties can actually be measured in sufficient reliability to use them for similarity assessments. So just to summarize how different organizations, specifically ECHA and EPA, use different properties for identification and grouping of materials. We have in the European definition size and surface area, and then additionally for the nanoforms size, shape, and coding. For the EPA reporting scheme, which is not yet fully in action but probably will be, it uses size and unique and novel properties, which is of course not easy to measure, let alone to agree what it really means. But then for discrete forms, they use size, shape, coding. That's completely the same as for the nanoforms. Additionally, surface area, dispersion, stability, surface reactivity, and charge. And I'll propose some methods how to do this in the rest of the talk. But for grouping, you need clearly more. You want to know something about dissolution, in vitro toxicity, dispersion stability, surface reactivity. And in order to screen for exposure, we want to know something about dustiness. Is a material easily releasing aerosols during handling? We want to know something about the releases. So this is my end of the introduction, and I would like to move now to one of the elements of our grouping framework and show you something about dissolution. I think everybody would agree that in order to test dissolution, you want to have a relevant fluid. That's okay. But even if you have that, you still need to decide how you incubate your material in that fluid. And these are the different options. You might just put it in a beaker and wait. That's okay. Particles will sediment and ions will be somewhere, will diffuse around. You may be concerned about inhomogeneities and add a stirrer, and so to homogenize the whole thing during the incubation. That's better. But then you might think that, well, ions can be transported under a physiological condition. They might not stay where they are generated. And so there have been attempts to use dialysis tubes so that ions can move out. That's one move towards a non-equilibrium situation. You can go further and use flow-by cells, where you hold the material here, ions diffuse again through a membrane and are then transported away. Or the most effective non-equilibrium testing, you flow through your system through a membrane that holds the particles, transmits the ions. None of this is new. I would like to highlight that the materials, the mineral fibers community has used this extensively in the 1990s. So I don't claim we invented anything new. We just adapted it to nanomaterials, and we think it's extremely useful. Actually, there is an OECD draft technical guideline that specifies this, and the World Health Organization has used all of these methods extensively for mineral fibers. And this is how it looked in their report. This is the metric of the dissolution. They call it K. K, see it above here, is a measure of nanograms ions released per square centimeter of particulates per hour. So it gives you a rate of dissolution. This rate is given here for different mineral fibers, glass wool, rock wool, stone wool, asbestos. And it correlates quite nicely with half-life times in vivo. And this is why this method has been so essential to classify mineral fibers. Based on their biopersistence, they are classified either as biopersistent and potentially carcinogen or not. And so this method might really be of use for nanomaterials too. So this is what it looks like in the lab. You have a fluid, you have flow through cells, several in parallel, and you sample whatever is eluted from the cells into this automated sampler, which we can program. And there are some details. I don't want to go into details now. We can have questions afterwards if you want to, but essentially we integrate all of the ion flux and we extract this K metric, so the dissolution rate for all different time points. And then we went a step further and compared this setup with a parallel setup that Alison Elder and Günther Oberderster run, and again one that is in another institute of our network, Utah. And the final result looks quite okay. So here, the solid line, this is the biokinetic study that Joe Bryan, who just entered the room, has done in these labs a couple of years ago. And this is the black line is barium sulfate. So they differ significantly. And now you see in the dashed lines our simulation in this dissolution setup. So we can clearly differentiate between these two materials, and we can have conditions that exactly mimic the in vivo dissolution rate. And now we apply this to many more materials, different versions of ceria. It matches the observation in vivo, essentially in absence of clearance. Barium sulfide, we finally understand this material which does dissolve under in vivo situations, and the soluble ones, copper oxide, zinc oxide, and those that are dominated by physical transport, the silicates, bentonite kaolins, so silicon aluminosilicates, or the iron oxides, which just do nothing. But interestingly, these materials also change morphology. So this is a silica before and after the dissolution. And if you make an automated evaluation of the EM scan, you see that the size shrinks. That's kind of what you expect. The material is dissolved, it shrinks. For barium sulfide, this is not the case. The material grows. After dissolution, the particles have become larger. Small particles, high curvature areas have disappeared. This is not completely new. This is something that is known as Ostwald ripening. So there is a marginal solubility of the ions which reprecipitate onto the existing crystals, and so they grow and they become more crystalline. And in fact, there are observations which I can show today, but the in vivo situation has the same phenomenon. Again, this effect is not unknown from the old studies on mineral fibers. So just to show you the analogy, by varying the flow rate in our dissolution setup, we essentially vary the equilibrium between the rate of ion generation and the rate of ion removal into the detection units. And so we shift the time that this material has to reprecipitate. So by scaling up the flow rate, we should see a higher dissolution, and that's what we see. So here we see surface area over flow rate. So if we go up to higher flow rates, this material has an steadily increasing apparent dissolution rate. This is not the case for something like copper oxide, which is up here. There is scatter, but essentially no dependence on the flow rate. But more than 20 experiments on barium sulfate all collapse into this one master curve if we scale it by the K metric and the flow rate. And this phenomenon is known from gel formation on mineral fibers. Here you see the same metric, the same parameters, and you see for different materials that they have the same dependence, that with increasing surface area over flow rate, SA over V, the apparent K goes down. And they related to gel formation on the surface of these fibers. That's a reprecipitation process, very similar to the reprecipitation of barium sulfate. Sorry for going into this detail, but barium sulfate was a difficult case. and it gives us much confidence into this assay that we can understand this material finally. We know that it reprecipitates into barium sulfate from XPS, not going into detail here, but it, unlike silver, for instance, which does sulfidation into a different species, it remains the same species. And so this is our final recommendation. For a simple assessment of solubility, the stirred beaker is quite okay. And actually, Health Canada has funded a study which published a very nice investigation on different zinc forms, which supports that this can be done. You just need to know which media to choose to be relevant, and this showed reproducible results. But for borderline cases, I think we need these. We need these flow through or flow by cells, and this is what we recommend for tier 2. But we can now go a step further and look at ecotox situations. So we had Daphnia medium, and we compare it against PSF, phagolysosomal simulant fluid, which was invented by Alex Stefaniak. And we look at two different versions of zinc oxide with a coating, that's the NM111, and without a coating. And interestingly, the coating does essentially nothing in the very acidic medium. There's a minimum retardation of just 2 hours or so, so that's not relevant. So coating does not prevent grouping for pulmonary fate. But the coating does have a significant effect in the ecological situation, where it takes much longer for the coated material to dissolve, and that's quite significant. So groups are not universal. They depend on the perspective. If you want to group for ecotox effects, that might be different groups than grouping for human tox effects. Finally, to compare the diversity of materials, different shapes, for instance, iron oxides, do not prevent grouping by their fate, because the dissolution, in that case no dissolution, is unaffected. Different sizes have not shown any significant differences for different cerium oxides, different organic pigments, different iron oxides, different coatings for the acidic medium, relevant for the lysosomal fluid, no differences. So there are some similarities that we can exploit for grouping. But of course, dissolution is not everything. We're also interested in physical transport. So we need to know something about affinity of materials. That is the next topic. How do we look at affinities and dispersion stabilities? There is an OECD draft guideline, which was really intended to be used for an ecotox situation, but we think it's quite useful and can be adapted. So this is the original protocol. It tells you, take your powder, bring it into a relevant medium, disperse, and then watch over, analyze over time how it reagglomerates and settles. And so different tests would be would show maybe different materials would show different dynamics, but essentially this draft guideline already proposes a grouping, because it says if over 6 hours less than 10% remain in the supernatant, then this is a low stability material. If more than 90% remain dispersed, this is high stability, and then two groups in between. I think this is quite useful. That's very pragmatic. They propose two different methods. You can use ICP-MS. That is, of course, nicely quantitative, but it's not so easy to have excellent time resolution with that. And it's also more expensive. They also propose UV-vis. So essentially, you have this cuvette here in a laser beam. And you can have higher time resolution and essentially the same dynamics. So we chose UV-vis for our investigations because we can automate it. We can have 7 or 8 cells in parallel on an automated UV-vis sampler. We can have good time resolution. And these are some selected results. In blue, protein-containing medium. In orange, eco medium, that is Daphnia magna medium, and you see that different forms of ceria or different forms of titania do behave differently. In physiological medium, this one is much more stable than this titanium. We still have to do the calibration of these results. Does that really prevent grouping? Should these be in different groups? We still need to look at the actual in vivo fate in detail and assign cutoffs and group ranges. But certainly for the ecological situation, it's quite easy. All of these materials go into the unstable group. So that gives us a very important hint. It tells us that aquatic testing is maybe not so essential. Sediment and soil should be much more in the focus for these materials. And then we use supporting methods, which are more quantitative, to look at dissimilarities, to understand why materials are different. And this has relations, of course, to Phil's beloved topic of dosimetry. Here is an even longer list of materials that we looked at in our nanogravure grouping project. And this is the D50 that we measure by analytical ultracentrifugation. And we also look at the predicted stability from the measured sedimentation rate by centrifugation and by Glenn Deloitte's method, which is based on an average effective density and an average size from DLS. Now, I'm sorry for this graph. It's hidden behind there. You should see on the graph that many materials, such as these iron oxides, have ranges that go far beyond a few micrometers. They can extend up to 100 micrometers. That's far beyond the scale that the DLS could see. So you tend to underestimate the spread of materials if you just look at DLS. That's one point to consider. We also see clear differences with different coatings. And so we think none of these methods is completely ideal. There are advantages of the OECD type method or of the AUC because they include all the heterogeneity. They measure distributions of settling dynamics. However, with the VCM method, we can assess quite easily the differences, and we can perform modeling that includes diffusion. AUC, we don't include diffusion in the modeling. So maybe none is ideal, but then again, the differences are not so dramatic except for this one material, and we all agree that this one has clear differences against the others. So we don't think that we need to invent something completely new here, but instead we try to use the OECD type for screening, and then for uncertain cases we would look into the details to assess dissimilarities or similarities of materials. But clearly, and that's an important point, if you just look at the hydrophilicity of these materials, you cannot predict their actual agglomeration potential in the relevant fluid. There are too many other factors that contribute, and that's one reason why the hydrophilicity is not useful for grouping. Now, let me come to the third box here, biophysical interactions and cellular effects. We used a lot the macrophage assay. That's Martin Wiemann's work at EBE Institute. This one example shows you the cerium dioxide without the macrophages, with the macrophages. And nice thing about this assay is you see how the cells take up the material. You know that they have been taken up. And the accuracy is quite good, 95%, compared to the in vivo situation. So this macrophage assay is one of the cornerstones of the tier 2 extrinsic properties. But maybe in some cases materials don't sediment, or you don't know if they are truly available to the cells. So these abiotic assays might be interesting. And we looked at 3 of them in more detail. Last year we finished the work on the cytochrome C assay, and that's published from Mathilde Delaval. It works, but only for the very reactive materials. It's not so sensitive, but it can be transformed into a useful assay. There's lots of evidence that the electron spin resonance is quite useful, but you still have to choose which spin trap you use. So essentially ESR is not ESR. there's a whole diversity of spin traps which test different reactivity mechanisms. So that's something to explore and to consider. And then we took up from the previous work from Dimitar Bello and others, and we looked in more detail into the FRAS assay. So here you incubate a material in human blood serum, then you remove the materials, you separate the particles out, and then you add a ferric complex that tests if the original antioxidants in the blood are still active. So you test the reduction of this iron complex. So it's a 3-step process, and it turns out to be very, very sensitive. We just published the detailed protocol on this. It's the first time that the protocol is described in full detail. And here are some selected results. We now do a concentration series. All of these have triplicate error bars. You just don't see it on them. You see it here. So different seria materials can now be differentiated. That was very important for us because we always see in vivo effects with seria, but we found it very hard to see any surface reactivity of seria. Now we have a measure that also differentiates different nanoforms. Manganese oxide as a positive control is very reactive. And then interestingly, we see that some materials just don't come to zero at low concentration. That's materials that dissolve. These are the effects from zinc ions. So the test itself will give us a hint if the reactivity is due to the particles or to the ions. Again, this is important because it helps us to rationalize findings. So we have 2 orders of magnitude between the negative and positive control. I don't want to go into details here, but it's kind of reasonable what we see. So for instance, differences between different forms of iron oxide that are linked to the surface structure. We see that a coating of zinc oxide reduces reactivity. And we see differences that can be linked to the availability of reactive species on the surface. So that makes kind of sense. But of course, you want to know how it compares to other assays. So here's a cross correlation between the FRAS and the ESR assay. We have 2 ways of evaluating FRAS, 2 ways of using spin traps in the ESR. I don't want to go into details. The key message here is that it's not a diagonal, which means that these assays are not interexchangeable, but they are complementary. They might be, one of them might be blind for one material, one of them might be very sensitive for another material. So potentially we will have to build a weight of evidence approach to assess surface reactivity, at least for something more complicated like ECHA dossiers. But then if I read the guidance on the EPA discrete forms, it just says, and very correctly so, you should use the same method when measuring the change in properties. That's exactly what I would do here. Just pick one and compare similarity of different forms. So again, methods are available and it can be done. However, none of this is really standardized or commonly accepted. That is certainly a challenge. To finalize on the toxicological side here, I want to show all of the results which we had from our inhalation studies. So all of these materials have been tested in the short-term inhalation study. And you see that we roughly group them into 4 groups based on the no effect levels. And it turns out that our grouping framework correctly assigns materials because we find that they either are above the high aspect ratio or above the composition or the surface reactivity cutoffs. So in this sense, for this set of materials, which we used as a training set, this is consistent. And of course, we now need to go further. We need validation sets, and that's exactly what we are doing now. We add more materials and see how this framework performs. One of them is different organic pigments, and just to highlight that one of them here, the red organic one, has also been in this house for testing. It was part of the incineration studies. And there are many more. There's a a large variety of materials. We know all their properties. And if we now apply them to our grouping scheme, then it turns out that for this red organic pigment, whether it's coated or whether it's nano or whether it's non-nano, they are all passive. And the same applies for two different sizes, nano and non-nano of the oranges. And there is something for the blue pigment, which is high enough to put it into the group of active materials. I'm sorry for that, it's this pointer. But it's not confirmed by in vivo, so this would be a false positive assignment. We can live with that. That's just one example. We now go further, and in the nanogravure project, we add many more materials. So this was the main part of the talk where we look at the hazard-related groupings. Now let's look at life cycle. You want to know if a material releases substantial aerosols or not. And one of the ways to do it is the dustiness index. There is a European standard for several methods, and I'm not going into details here, but some of these methods have a challenge that they require hundreds of grams of material for testing. That is prohibitive for research materials, and it can also be a challenge for some conventional materials. So we need something that works with lower masses. And I just want to mention that I'm quite happy that we could recently join the Danish guys in SCWE and the French Institute ENS. We now also operate a small rotating drum, and this is generating useful materials in the previous studies from Kjell Jensen and colleagues who developed this technique. So we hope that we can soon follow up on that and hopefully establish this method as an industrial standard for us. They established 4 decadic groups of dustiness. It's an input parameter for exposure modeling. That's very interesting. So it enables you to model for instance, the distribution of particles in this room, keeping track of all the ventilation, etc. And it is one descriptor of granulometry in reach. These are examples, again, not from us. That's in this OECD report, which essentially is from Kjell Jensen. The titania, NM105. You have a inhalable and respirable dustiness index, which is clearly differentiated here from a silica material. So that's something you can use. Tells us how much milligram of dust being generated per kilogram of handled material. But of course, dust is not everything. Many materials are more complex. Let me just show you 2 examples. If you should have a cooktop panel, Saran, this is a nano-enabled product. Why? Because the glass melt is adjusted such that as it melts, it generates nuclei, and these are specific oxide nuclei in actually quite a high concentration, 10 to the 24 per cubic centimeter. And this is essential to prevent the thermal expansion of the glass. So this makes it a zero or low thermal expansion glass. And our collaborators here from Schott, they like to claim that this is the largest nano-enabled product ever made, 8.6 meters diameter. This is a mirror for an astronomical telescope, which of course needs to be very precise and is not allowed to expand. They use exactly this material for the purpose, and that's in this book chapter. And so we call these nano-enabled products because a nanostructure is essential for the final functionality. Does not necessarily mean that nanomaterials, in the sense of particles, are added anywhere in this process. Another example are aerogels. Aerogels are a reactive process. You have a reaction that you lead in a way that finally you have a porous, a foam structure, which is structured on the nanoscale. It does not necessarily include the use of particles. So if we want to assess safety of such materials, we need to know what they release during the handling and the use of these materials. There's a nice framework for that. We published a similar scheme earlier, and then Tim Duncan from FDA converted it into this very nice scheme that I almost fully agree with. Starting from an intact nanostructured material, you can have mechanical forces, and then depending on the mechanical properties of the matrix, if it's a tough matrix, you will have these sorts of fragments. Brittle matrix tends to have more protrusions of nanomaterials on the surface. The same thing would apply for the nano-enabled products. And chemical forces, of course, have completely different effect because they change the composition of the matrix. They can degrade the entire matrix. Thermolysis, hydrolysis, photolysis all have their action. And at the time of writing this, Tim had obviously some uncertainty about thermal decomposition. Well, that's one reason why we keep investigating this with Phil here. I think we're making good progress on that. So this might actually not be true, but the question is, how can release properties be used as a grouping hypothesis? We need methods to do this. And so in the nano release project, together with many partners, especially here in Canada and in the US, we established this protocol. We use, and that's important, an ISO standard to degrade the material. So we know that this is relevant because people use it all the time for product durability testing. And then we just add sampling by shear forces, and we analyze these materials by different methods. And all of this is published in Carbon earlier this year. And actually, from EPA and BASF and National Research Center in Canada, all of the results, the raw data, is available on the NIST servers. So at the Materials Genome Initiative, you can look at everything. This is the result when you now expand to different materials. In the nano release, we looked at this epoxy and this polyamide. They were clearly different. And now we took a bunch of materials and I color-coded them here by the matrix. So all of the cements have this light blue, polyurethanes have this dark blue, and polyamides have this gray scale, etc. Obviously, they cluster by color. It tells us that across many orders of magnitude, from polyethylene to epoxy, the matrix dictates the rate of release, and there is a modulating effect by the nanomaterials. I keep struggling with this pointer, I'm sorry. We have a modulation. That's important to say. The carbonaceous materials are black, they tend to passivate. And of course, photocatalytic properties from zinc oxide tend to accelerate degradation, etc. That's a modulation, but it's only a modulation. The key grouping is by the matrix. Let's look at mechanical properties. This is a setup that we use in our labs. It's essentially a small clean room, particle-free air. We have a drilling machine that we can also use as a sanding machine. And here, my postdoc, Nicole Neubauer, did a very nice work on comparing different conductive plastics. And specifically, she chose 3 materials that have the same functionality. Well, functionality doesn't mean same filler content, because in order to decrease resistivity by 11 orders of magnitude from the pure polymer, you either add 3% of carbon nanotubes or 5% of graphene or 10% of carbon black. That's exactly one of the potential advantages of carbon nanotubes, that you would have to add less. Unfortunately, I have to tell you, this material is still substantially more expensive than this, so nobody will buy it, and we don't sell it for that reason. But if you look at release now, these materials are not so terribly different. There is no very specific effect, and Nicole did this nice comparison. It turns out that the maybe I shouldn't even use the pointer here. The tensile strength is key to predict the releases So that gives us another property that we can use for nano-enabled products. Tensile strength tells us how to predict release rates, and not so much the functionality, like conductivity, and not so much the shape or substance of the nanofiller. These are secondary criteria. So are these fragments toxic? And we picked a metal, copper, that is known to be toxic, and we looked at copper phthalocyanin pigments in automotive coatings. So you have the blue coating, you see this blue dust here from sanding. We looked at size distributions, and I just want to highlight here that we then did cytotoxicity. We do have an effect from the ion controls, which we adjust to be the same copper content as in the fragments that we look at from the coatings. So this has the same copper content, but in fact, we don't see an effect from the fragments. We do see an effect from the pure materials, but not from the fragments. And this was work from Shengfang Pang, a postdoc published in Nano Impact now, was just accepted. And that's just one study that adds to many previous studies from the Nano House project, again from the NRCWE, that looked at different polymers with titania, silica, or CNT fillers, including in vivo studies, and they confirmed the same conclusion that the toxicity is only weakly, if at all, modulated by the presence of the nanomaterials in these fragments. Now, I went one step further. How am I doing with time, Phil?
We'll give you like 5 minutes.
That's fine, thank you. Ecotoxicity. Nobody really looked at that. And so in the Sun project, we took the example of a car bumper. This is a part that you might find under an Alfa Romeo Mito, because we did this in collaboration with the guys who do that for the Fiat affiliation. And so we compared the synthesis phase, the formulation phase, the use phase. These are these kinds of fragments then finally that we apply to different organisms. This is Enchytraeus crypticus, so a worm that lives in the soil, and this is a zebrafish. And to walk you through this diagram, we look at these different stages of the life cycle. Synthesis, pure nanomaterial, then you see the ENM content goes down from 100% to just a couple percent, depending on the application. So this is the same product made with different materials, organic pigment, iron oxide, or CNT. And you see how over the life cycle, the iron oxide content is the only one that remains at 100%, because that's a value that I took from our incineration studies here. It remains, we know that it is again 100% at the end, but 0 if we assume waste incineration for CNT or organic pigment. And we can do the same for size, and we can do the same for surface chemistry, XPS, hydrophobicity. When we look at cytotoxicity in rainbow trout cells, we look at the toxicity in the earthworm, Enchytraeus crypticus. We always find an assimilation of the nano-enabled fragment properties to the matrix. In the pristine synthesis phase, these nanomaterials are completely different from each other. But if we look at the fragments that are released from a product, they behave all the same because properties are dictated by the matrix. Now we did something different, just to confuse you a little. We took one nanomaterial and put it into different matrices. So CNT in cement, and polyoxymethylene in epoxy, and in polypropylene. And in that case, we have a diversification of properties because, again, this material assimilates to the matrix. So what's originally a very hydrophobic material dominated by carbon here then assimilates to the matrix if we measure the fragments from the use phase. And again, we look at zebrafish. Andrinel did that. They looked at Enchytraeus crypticus. Monica Amorim did that. The rainbow trout cells, that's Jose Maria Navas. and we looked at soil and at sewage sludge. Kerstin Hundrinke did that, and this confirms this hypothesis that the matrix dictates the behavior. Of course, there are some uncertainties in this because it's really an exploration into something new, but we are writing this up now, and we hope that it will generate a discussion on how to use life cycle as a grouping criterion. So to summarize the life cycle, first look at the process. Understand what process you're looking at, what is your scenario. Then you know already the relevant medium that you would use for dissolution or hazard testing. And you know the metrics of release and exposure. Second, look at the matrix. We think a big progress in terms of similarity assessment can be made if you just know if your specific material that you want to investigate has a matrix that is similar to a product that you have investigated before, because it will tell us a lot about the rate and toxicity of fragments. And finally, the modulation, which is scientifically very interesting, but maybe in a regulatory sense, not the most important, that's the modulation by the nanomaterials. And I think the most interesting that we found so far are the catalytic effects, which we see in incineration, and we also see catalytic effects in UV degradation, because again, the specific properties can contribute a lot there. So to finalize, this ECETOC scheme, we think, is quite efficient to sort out the big three groups that can be easily read across to existing data. That's the soluble materials, the high aspect ratio materials, and the passive materials. The properties that we use are consistent with those properties proposed by the ECHA framework. Additionally, grouping by matrix for nano-enabled products is really a way to simplify the assessment of the life cycle generated fragments. We currently expand this. Much of what I told you is from the nanogravure project, and we will have something similar for environmental grouping. Methods and benchmark materials are increasingly available, and we are now fine-tuning the cutoffs and group ranges. I have to show this as very last slide. I know the metaphor is old, but it really looks like an iceberg here. Bigger at the bottom, this is the production of particulate materials, not specifically nano, just taking the global production of typical fillers and pigments. And you see in gray those materials which have been used a lot for release studies, not overall nanotoxicity, but release, because I had this data from Stephen Frudgett's paper. And you see there's kind of a mismatch. We know a lot about CNT and silver, but we should know much more about these kinds of materials. And that matches the finding that we have from those reporting schemes that in Europe are now mandatory, and with the EPA are probably coming in the US too. And there's the Canadian scheme already. And the European Commission now acknowledges that the experience from the existing notification system in France concerns innovative nanomaterials only to a small extent. Rather, it relates to materials which have been on the market for a long time and which companies were forced to assess against the nanomaterial definition for the first time. I'm not saying this is necessarily bad, but this is the case. We are currently re-evaluating conventional materials with respect to their nanoscale properties. And I can only ask you from the academic community to join us in investigating these more conventional materials, because clearly there is a regulatory need and a regulatory request to know more about these based on the knowledge that we gained looking at the innovative materials. And with that, I want to thank all of our collaborators. None of this would have been possible with BASF alone. It is all from collaborations, and specifically I would like to highlight partners from the nanogravure project, because this was the source of much of the data that I showed you today, and the Sun project, which was the source of the ECOTOX investigations. And with that, I thank you a lot for your attention.
Comprehensive presentation. We're running behind schedule, but we will start with our guests from online. So we have any questions, Alison? Okay. So then we should switch to the audience. If there are any questions from the audience. I have a question, which is it's more actually it's not it's a comment rather than a question, but I want to emphasize the importance of looking on synergistic effects, because we know that in biology 1 + 1 usually is not 2, it can be 3 or 4, and we publish 2 papers now, which I think they make very strong case. Actually, one is you mentioned it is the EST published paper, is with the incineration, when you have catalytic nanoparticles and you have all these SVOCs emitted at the same time, come up with some transformations and conversions from low to high molecular weight gauges on the particles, which can induce toxicity. And the other paper we just submitted is for another interesting nano-enabled product, is the toners used in printing equipment. Actually, Sandra just submitted that paper, which we found exactly similar synergistic effects, and so I think it should be part of the grouping that we need to take into consideration that in most cases and scenarios, you may have some other co-pollutants emitted at the same time, and they may result to these synergistic effects that can amplify the toxicological profiles of the releases. So I want to comment on this and see how you see this fit in
well, that's a nice challenge that you're putting up there. However, currently, I must really say we focus on complying with the regulatory schemes that are being put up, and they are of course substance-based. For a specific scenario, I agree, synergistic effects need to be looked at, but this scheme really is targeted at finding similarity on substance basis. Yeah. And this is what is required for risk assessment.
Okay, thank you. Any more questions from the audience?
So was there a particular rationale as to why just a macrophage was chosen as a cell type of choice and not other cell types, for example, epithelial endothelial cells? Because when you read through papers, there are studies which focus on those individual cell types. So what's the reason? Is it just that you were looking at something like lung as a might be taking most of the particles and hence macrophages and not some other tissue type?
Beginning but the end of a project to choose this specific cell type. So we did look at many other cell types, and we included also co-culture models. In the end, the selection is based on the accuracy as compared to in vivo, and you're right that this model, this framework was developed really with an eye towards pulmonary effects, because that was, let's say, the key focus for occupational safety concerns. It might be true that if you have, for instance, an oral scenario, although oral effects with nano are generally not so pronounced, you might look at other cell types. So that's something that we probably might want to specify out in more detail. But the choice of this specific macrophage is really because it has such a good productivity against the inhalation studies.
All right, I think we need to stop it here. Thank you again for the wonderful presentation. Thank you all for attending, and look forward to seeing you in October when we will start the new Nano Lecture Series for 2017. Enjoy the summer. Bye.